Hello students, a very good morning to all of you. So tell me, my audio is clear and my screen is visible to everyone? Okay, all right. In today's class, we are going to discuss a brief about the human reproduction chapter because you have already done this chapter in your school. So in today's class, we are going to discuss this chapter in brief, and in the next class, we'll do the problem solving of this chapter. So first we are going to discuss is the male reproductive system, which is located in the pelvis region. And the male reproductive system, it consists of the testis, which is the primary sex organ. Why it is the primary sex organ? Because first, it produces the male gamete, and second, it produces the hormone, which is androgen or testosterone. Male reproductive system also include the accessory ducts, glands, and external genitalia. The testes, they are present in pairs and they are located or present in a pouch-like structure which are called as scrotum. And these scrotum, they act as a thermoregulator. They maintain the low temperature of the testes. They maintain 2 to 2.5 degrees Celsius temperature lower than the normal internal body temperature. And this temperature is necessary or mandatory for the spermatogenesis. That is why the scrotum, they are also known as thermoregulator. So in adult, the testes, they're oval in shape. They have 4 to 5 centimeter in length and width of 2 to 3 centimeter. The testes, they're covered with a dense covering, and each testis has a 250 compartment which are known as testicular lobules. Remember this very important point that each testis, it consists of 250 testicular lobules, and one testicular lobule consists of 1 to 3 seminiferous tubules. You can see in the image, these are the seminiferous tubules. So in one testis, there are 1 to 3 seminiferous tubules are present, and seminiferous tubule is the site where the spermatogenesis takes place. That means the production of the sperm, it occurs in the seminiferous tubules of the testis. In the seminiferous tubule, there are two types of cells are present. One is the male germ cell, which are also known as spermatogonia, and the Sertoli cell. So the male germ cell, these are spermatogonia, which are going to give rise to the sperm after the mitotic division. And Sertoli cell, their function is to provide the nourishment to the sperm. Now, outside these seminiferous tubules, there are tissues are present which are known as interstitial tissues. And in the interstitial tissues, there are some cells are present which are also known as interstitial cells and the Leydig cell. The function of these Leydig cell or the interstitial cell is to produce the hormone which is the androgen or testosterone. Apart from these Leydig cell, in the interstitial tissue, there are some immunologically competent cells are also present. These cells, they are like a leukocyte. They are providing the protection against the bacteria, microbes, or virus. Now, after the testes, there are accessory ducts are present, which includes the retitestis, vasa efferentia, epididymis, and vas deferens. So student, remember from the seminiferous tubule, I told you now one testicular lobule, there is 1 to 3 seminiferous tubules are present. Right? And from each seminiferous tubule, there are some straight tubules come out or the tubes come out, and these tubes are known as tubular recti. So if this is the testicular lobule 1, These are the seminiferous tubule, and from these seminiferous tubule, the small ducts will come out, and these ducts, they are known as tubular recti. And these tubular recti, they will combine and they will join a common duct, which is known as rete testis. Now, these rete testis, they will open into vasa efferentia. And vasa efferentia is the one which leaves the testis. And this vasa efferentia will open into epididymis. And epididymis will lead into the vasa deferens, and vasa deferens and the duct from the seminal vesicle, they combine and they form a common duct which is known as the ejaculatory duct. Now this ejaculatory duct, it opens into the urethra. And the urethra originates from the urinary bladder. It extends through the penis to its external opening, which is known as the urethral meatus. So there's a single opening in case of male for the urine and for the semen. Remember this sequence, student. This sequence is very, very important. Many times this question came in NEET on the sequence of the accessory duct. So it's very important, remember the sequence. And please remember that vasa efferentia is the one which leaves the testis. Now moving to next, the male external genitalia is the penis. And the enlarged end of the penis is called the glans penis, which is covered by these folds of skin called foreskin. There are three accessory glands are present. One is the seminal vesicle, prostate gland, and the paired bulbourethral gland. Remember, the seminal vesicle is in pair, prostate gland is single, and bulbourethral gland is also present in pair. Remember, this is important. This is the seminal vesicle gland, this is the prostate gland, and these are the bulbourethral gland. And all these glands, they perform a very important function in contributing the seminal plasma. So semen is consists of the sperm and the seminal plasma. Sperm is contributed by the testis, and seminal plasma is contributed by the accessory glands. And accessory glands include the seminal vesicle, it includes prostate and bulbourethral. So bulbourethral gland, it contributes in producing the mucus, and mucus help in the lubrication of the penis. Seminal vesicle gland, it contributes in the fructose. Prostate gland, it contributes in calcium and certain enzyme. Okay, so remember this. Fructose by seminal vesicle, calcium and certain enzyme by prostate gland. So seminal plasma is rich in fructose, calcium and certain enzyme. Remember this, very, very important. Now, next we are going to discuss is the female reproductive system. So female reproductive system, it consists of pairs of ovary. Ovary are the primary sex organ because it produces the male gamete, that is ovum, and also the hormones, which are estrogen, progesterone. Okay. Apart from the ovary, the female reproductive system is also consist of the oviducts, which are known as the fallopian tube, the uterus, vagina, cervix, and the external genitalia. The ovaries, they are located on each side of the lower abdomen. It is about 2 to 4 centimeters in length, and it is located in the lower abdomen and connected to the uterus wall or the pelvic wall by the ligaments. Each ovary is lined by the epithelium, and inner to the epithelium, there are stroma tissues are present. And these stroma, they are divided into two zones. There's a outer one is known as the peripheral cortex, and the inner one is known as the medulla. So remember this, Very, very important point that the stroma is divided into two zones. If this is the ovary, this middle portion is known as the medulla and the outer is known as the cortex. Ovarian follicle, the primary follicle, secondary, tertiary, Graafian follicle, all the follicles, they are present in the cortex region. Remember this, very, very important point. So fallopian tube is 10 to 12 centimeter long. It extends from the periphery of the ovary and to the uterus. So fallopian tube, it consists of three parts. There's infundibulum, ampulla, and isthmus. The part of the fallopian tube which is closest to the ovary, this is the ovary. And the part of the fallopian tube which is closest to the ovary, this is known as infundibulum. Very, very important. Remember this. Which part of the fallopian tube is closest to the ovary is infundibulum. And infundibulum, it has a finger-like projection, and those finger-like projection are known as fimbriae. So these finger-like projection, they help in capturing or grabbing the egg once it is released during the ovulation. Now this infundibulum, it leads to a wider part of the oviduct, which is known as ampulla. And ampulla is the site where the fertilization takes place. This is also important, that in which part of the fallopian tube the fertilization takes place, the answer would be ampulla. Remember this, very, very important. Now, the third part of the fallopian tube is the isthmus. Isthmus has a very narrow region, and this is the part of the fallopian tube which connects the fallopian tube to the uterus. Next is the uterus, which is single and it's also called as the womb. The shape of the uterus is inverted tear. Uterus, it consists of three parts. The upper part is known as the fundus. The middle is known as the body, and the lower part of the uterus is known as the cervix. Uterus, it consists of three layers. The outer layer is known as the perimetrium, which is the outermost layer. Middle layer is known as myometrium, and myometrium consists of smooth muscle. Myometrium is the layer of the uterus which shows the contraction during parturition. So remember this very, very important point, that which layer of the uterus consists of smooth muscle. Your answer should be myometrium. Now, inner to the myometrium, endometrium is present, which is the innermost layer of the uterus. And the endometrium is the one which undergoes the cyclical changes during the menstruation. Remember this, that which layer of the uterus undergoes these cycle changes during the menstruation? Your answer should be endometrium. And there's one more thing. Inside the endometrium, there are two layers. One is stratum basalis, one is stratum functionalis. So stratum basalis remain constant, but stratum functionalis is the one which undergoes the changes. Okay? So out of three layers of the uterus, endometrium is the one which is undergoing the changes. And inside the endometrium, stratum functionalis is the one which is undergoing the changes. Remember this, very, very important point. Now, the female external genitalia includes the mons pubis, which is a cushion of the fatty tissue covered by the skin and the pubic hair. Inner to the mons pubis, there are labia majora are present, which are the fleshy folds of the tissue. Inner to the labia majora, there is labia minora are present, which are also the folds of the skin. An opening of the vagina is covered by a membrane, which is known as the hymen. And at the upper junction of the two labia minora, or above the urethral opening, there's a finger-like projection is present, which is known as clitoris. In female reproductive system, there are two accessory glands are present. Number one is the Bartholin gland, and the another one is the Skene gland. Skene glands, they are present around the urethral opening, and the Bartholin glands, they are present at the vagina. Next, we are going to discuss is the mammary glands. So mammary glands, they are the paired structure. They consist of 15 to 20 mammary lobes. And each mammary lobe is filled with the alveoli. And the cells of the alveoli, they secrete milk, which is stored in the cavity of the alveoli. And from these alveoli, let's say this is the mammary lobule. Inside the mammary lobule, alveoli cells are present. And from these alveoli cells, the small tubes will come out. And these tubes, they are known as the mammary tubules. These mammary tubules, they combine and they form a common tube, which is known as the mammary duct. Now, several mammary ducts, they combine and they form a wider portion, which is known as the mammary ampulla. Now, this mammary ampulla, they will connect to the lactiferous duct, through which the milk is sucked out. So in this phenomena, there are two hormones are involved. One is prolactin and another one is oxytocin. Remember one very important point, like many people, they answer it wrongly. Remember this, that oxytocin is the one which is involved in the milk ejection. And prolactin is the one which is involved in milk synthesis. Very important point. If you ever get a question that which hormone is involved in the milk ejection, your answer should be oxytocin. And which hormone is involved in milk synthesis, your answer should be prolactin. Okay? Because when the oxytocin is released, oxytocin main function is in the contraction of the muscle, right? So when the oxytocin is released, oxytocin, it starts contracting the muscles around the lactiferous duct, because of which the lactiferous duct squeeze out and the milk come out. Next, we are going to discuss the gametogenesis, and first we are going to discuss spermatogenesis. As the name suggests, spermato, and genesis means formation. So, spermatogenesis means the formation of the sperm. I told you that the sperm formation takes place in the seminiferous tubule. In seminiferous tubule, there are two types of cells are present, germ cell and the Sertoli cell. Germ cell are the one which are going to give rise to the sperm. Main function of the Sertoli cell is to provide the nourishment to the developing sperm. So the germ cell, they are known as spermatogonia. Now these spermatogonia, they will increase in size or undergo growth. Now they are known as, after growth, these spermatogonia, they are known as primary spermatocyte. Now these primary spermatocyte, they will undergo the mitotic division. They will undergo the first mitotic division and will produce the secondary spermatocyte. They will undergo the second mitotic division and will produce the spermatids. Remember this sequence is very important. Spermatogonia, primary spermatocyte. Remember, this is diploid. This is diploid. This one is haploid and this one is also haploid. Now, once the spermatids are formed, now these spermatids, they undergo the transformation or the differentiation and they give rise to the sperm or the spermatozoa. This process is known as spermiogenesis. Okay, and when the sperm, they are immature, their head are embedded in the Sertoli cell. Once the sperms, they are mature, they get detached from the Sertoli cell, they lie in the lumen of the seminiferous tubule and they are released. Once they are ready, they are released from the seminiferous tubule. And this process is known as spermiation. Okay, so these three terms are very, very important. Okay, very important. Many times this question comes. You should remember the difference between spermatogenesis, spermiation, and spermiogenesis because these three terms, they have a different meaning. Spermatogenesis means formation of sperm. Spermiogenesis means when the spermatid, they are transformed into spermatozoa or sperm. Spermiation means when the sperm, they are fully mature and released from the seminiferous tubule. So these three terms, they have a different meaning. Remember this, student, very important. Now in spermatogenesis, the hormones are also involved. One is the gonadotrophin releasing hormone. This hormone is released from the hypothalamus and this hormone acts on the anterior pituitary. It stimulates the anterior pituitary and allow it to release the two hormones, which is LH and FSH. LH is the luteinizing hormone. FSH is the follicle stimulating hormone. LH will act on the Leydig cell and allow the Leydig cell to release the hormone, which is an androgen or testosterone. Remember this, very, very important, that LH will act on the Leydig cell and FSH will act on the Sertoli cell. So when FSH act on the Sertoli cell produces some types of factor. And what are those factors? They are ABVP. ABP are the androgen binding protein. Function of this ABP is to bind with the testosterone and increase the level of the testosterone in the seminiferous tubule. Because testosterone, it stimulates spermatogenesis. So function of androgen binding protein is to bind with the testosterone, increase the concentration of the testosterone in the seminiferous tubule so that to increase the spermatogenesis. Sertoli cell also release one hormone which is known as inhibin. And this hormone is released when there is an excess of spermatogenesis. When the spermatogenesis is taking place in excess, in that time, the Sertoli cell will release a hormone which is known as inhibin. And as the name suggests, inhibin. So it will act as an inhibitor. So this hormone will send the negative feedback to the hypothalamus and the anterior pituitary. When this hormone release gives the negative feedback to these two glands, they will produce a low level of LH and FSH, because of which the spermatogenesis will suppressed or reduce. Similarly, here also, when testosterone is produced in a very large amount or the spermatogenesis is taking place in excess, that time also the testosterone, it gives a negative feedback to the anterior pituitary and hypothalamus. So that a low level of testosterone would be released from the Leydig cell and spermatogenesis will suppressed. Remember the roles of the hormone, that on which cell the LH is acting, on which cell the FSH is acting. Now let's discuss the structure of the sperm. Sperm, it composed of a head, neck, middle piece, and a tail. Head is consist of two things. Number one is the acrosome, which is a cap-like structure. And inside the acrosome, the hydrolytic enzymes are present. And these hydrolytic enzymes, they allow the sperm to penetrate or to pass through the secondary oocyte. Head also consists of the haploid nucleus. Neck, it consists of the two centriole. There's a proximal centriole and distal centriole. Proximal centriole is involved in the zygote cleavage and distal centriole is involved in the formation of the tail. The exoneme of the tail, the filament of the tail is formed by this distal centriole. Role of the proximal centriole will become more clear when we will discuss the fertilization. Middle piece is known as the energy chamber. Why it is called the energy chamber? Because it consists of the mitochondria. And these mitochondria, you can see these are spirally arranged like this. And the spiral arrangement of the mitochondria is known as the Nibunkan sheath. Last part is the tail, and the tail helps in the movement of the sperm. It provides the motility to the sperm. Next, we are going to discuss is the oogenesis. In case of male, the spermatogenesis, it occurs at the puberty when there's a significant increase in the level of the gonadotrophin hormone. But in case of female, the oogenesis is initiated during the embryonic development stage. And during the embryonic stage, millions of oogonia are formed within each ovary, and no more oogonia are added after the birth. And these oogonia, they undergo the mitotic differentiation and form the primary oocyte. The primary oocyte undergo the first mitotic division. But this first mitotic division is not completed. This primary oocyte get arrested in the diplotin stage of the prophase 1 of meiosis 1. And when these primary oocytes, they are surrounded by the granulosa cells. Another name for the granulosa cells are the nerve cells or the follicular cells. They are the stromal cells only. So when these primary oocytes, if this is the primary oocyte, they're surrounded by the single layer of the granulosa cell. This is known as the primary follicle. So at puberty, a female has only 60,000 to 80,000 primary follicle. Rest degenerate during the phase from the birth to puberty. Now these primary follicle, they undergo changes and form the secondary follicle. The secondary follicle has more layers of the granulosa cell. I told you the primary follicle is having a single layer of the granulosa cell, right? Secondary follicle, they have more layers of the granulosa cells. And outside the granulosa cell, this theca layer is present. So secondary follicle, it consists of the many layers of the granulosa cell. It consists of theca cell and the primary oocyte. Remember this, that in secondary follicle, it still consists of the primary oocyte. That means the meiotic division, the first meiotic division is not completed. That means the primary oocyte is still arrested in the diploidine stage of prophase 1 of meiosis 1. Now this secondary follicle is transformed into the tertiary follicle. And this tertiary follicle, it has a cavity which is known as the antrum. And theca, which is formed in the secondary follicle, it gets differentiated in the tertiary follicle as theca externa and theca interna. Now, one very, very important point that in tertiary follicle, the primary oocyte completes its first meiotic division and produces the secondary oocyte and the first polar body. So in tertiary follicle, it has the secondary oocyte, it has antrum, which is the cavity, it has granulosa layers, it has theca, which is differentiated into externa and theca interna. Remember this point, that tertiary follicle is the one which has the secondary oocyte. And the secondary oocyte, it has a bulk of the cytoplasm, which is contributed by the primary oocyte. And the first polar body, it's later get degenerated. Why it get degenerated? Because most of the cytoplasm of the primary oocyte is contributed to the secondary oocyte. So first polar body is having a very low amount of the cytoplasm. And you know that most of the nutrients, they are present in the cytoplasm, right? So if the first polar body is having a very low amount of cytoplasm, that means a low amount of nutrient, because of which the first or the primary polar body does not survive for long. So it's after sometimes it get degenerated due to the low amount of the cytoplasm. Now this tertiary follicle, when mature, that means the cavity, the antrum cavity which is formed in the tertiary follicle, it increases in size. So the tertiary follicle get mature. Now this mature tertiary follicle is known as the Graafian follicle. And the secondary oocyte, it forms a new membrane which is known as the zona pellucida. And this zona pellucida, it is made up of the glycoproteins. Remember this point, very, very important point, that zona pellucida is made up of the glycoproteins. Now on the day 14, this Graafian follicle rupture and secondary oocyte is released from the ovary. And this process is known as ovulation. As you can see in the image, the Graafian follicle rupture and secondary oocyte is released. This process is known as ovulation. And rupture Graafian follicle, it forms a yellow color body which is known as the corpus luteum. Later, this corpus luteum also get degenerated and forms corpus albicans. Next, we are going to discuss is the menstruation or the menstrual cycle. The reproductive cycle in the female primates is called the menstrual cycle. In human, the menstruation is repeated as an average interval of about 28 to 29 days. And the cycle of events from one menstruation till the next one is called the menstrual cycle. So menstrual cycle, it consists of the three phases. There's a menstrual phase, there's follicular phase, and there's luteal phase. So first phase is the menstrual phase where the menses or the bleeding takes place for 3 to 5 days. So during this phase, you can see the level of the progesterone is very low. Remember, the main function of this hormone progesterone is to maintain the endometrium. So if the level of the progesterone would be very low, that means the endometrium would not be able to maintain. So shedding of the endometrium takes place when there's a low level of progesterone is present. And shedding of the endometrium leads to menses. And the phenomena is known as menstruation. Now, after the 5th day, there's a follicular phase started, which is also known as the proliferatory phase. During this phase, the follicle development takes place. That means from the primordial follicle to primary follicle, then secondary follicle, tertiary follicle, and then Graafian follicle. So during this follicular phase, you can see the level of the two hormones, which is contributed by the pituitary, that is FSH and LH, start increasing. Because FSH is involved in the follicle stimulating hormone, as the name suggests, follicle stimulating. So its role is involved in the follicle development from primary, then secondary, then tertiary, then Graafian follicle. So the level of the FSH start increasing during this follicular phase. Now, when the level of when the follicle start developing during this follicular phase, these follicles, they also contribute or produce the estrogen hormone. So you can see this in the image that the level of the estrogen is also increasing because these follicles, they start producing the estrogen hormone. So the level of the estrogen start increasing. And this estrogen hormone, it gives a positive feedback to the LH hormone. So on the day 14, these estrogen, it gives a positive feedback to the LH hormone, and the LH hormone suddenly increase. So you can see on the day 14, there's a sudden increase in the LH hormone. This sudden increase is due to the positive feedback from the estrogen. And when there's a sudden increase in the LH hormone, what happens? The Graafian follicle rupture, secondary oocyte is released. This process is known as ovulation. So ovulation is taking place on the day 14, which is also known as the ovulatory phase. Now secondary oocyte is released. The rupture Graafian follicle will form the yellow color body, which is known as the corpus luteum. And why this body is yellow in color is because of the lutein pigment, which are yellow in color, which impart the yellow color to this body. Now, corpus luteum function is to produce the hormone, majorly the progesterone. It also produces a small amount of estrogen, but majorly it produces the progesterone. So this corpus luteum start producing the progesterone. As you can see, now the level of the progesterone increases. And one more important point which I want to tell you here, when the level of progesterone increases, it gives a negative feedback to the anterior pituitary. So remember this point, the high level of progesterone, it gives a negative feedback to the anterior pituitary. So if it gives the negative feedback to anterior pituitary, that means the level of the LH and the FSH, which are produced by the anterior pituitary, will start decreasing. So you can see the level of FSH and LH start decreasing because there's an increase in the progesterone. Now, one more important point. LH hormone, it performs two functions. One, it is involved in ovulation, or it induces ovulation. Second, it maintains corpus luteum. Just now I've told you that the high level of progesterone gives a negative feedback to anterior pituitary and reduces the level of LH and FSH, right? So when the level of LH start decreasing, that means this corpus luteum, which is formed, is to also start decreasing. It also start degenerating. So the corpus luteum start degenerating, and this regressed corpus luteum is known as the corpus albicans. And corpus albicum does not produce progesterone. I told you that corpus luteum produces the progesterone hormone, and progesterone is involved in maintaining the endometrium. But progesterone gives a negative feedback to the anterior pituitary. Level of LH drop. And if the level of LH drop, this corpus luteum start degenerating and forming the corpus albicum. And corpus albicum does not produce progesterone. So you can see the level of progesterone drops. And when the level of progesterone drops, the endometrium, which was well developed, starts shedding. And when the endometrium shed, it leads to menstruation. So this is the beginning of the next cycle. I hope it is clear to everyone. So remember the role of the hormone, what are the changes that is taking place in the membrane of the uterus, that is endometrium. What are the changes that are taking place in the ovary, and the role of the hormone. This is important. Now let's come to the next topic, which is fertilization and implantation. So before I proceed with the fertilization, I want to discuss the structure of the secondary oocyte. The cells, which are, these are the corona radiata, these are the granulosa cells only. So the cells which are present outside the zona pellucida is known as the corona radiata. And the cells of the corona radiata, they are loosely arranged. Like this. They're loosely arranged. And these cells, they are cemented by the polysaccharide, which is known as hyaluronic acid. Now, inner to the corona radiata, there's a zona pellucida is present, and zona pellucida is made up of the glycoprotein. Remember this. Now, inner to the zona pellucida, there's a perivitelline space is present. This is the space between the zona pellucida and the plasma membrane of the secondary oocyte. Now, inner to the plasma membrane of the secondary oocyte, there's a cortical granules are present, which are present in the cytoplasm of the secondary oocyte. Now, when the sperm it come in contact with the secondary oocyte, the sperm it has to penetrate or break or pass through the three barriers. Number one is the high cement, which is holding the two corona radiator cells together, that is hyaluronic acid. So for that, the sperm has a cap-like structure known as acrosome, right? In the acrosome, there's a hydrolytic enzymes are present. And there are three hydrolytic enzymes. There's a hyaluronidase enzyme, there's zona lysine, and there's corona penetrating enzyme, right? So the first barrier that the sperm has to cross is the hyaluronic acid, which is cementing the corona radiata cell. So for that, the sperm has the enzyme, which is hyaluronidase enzyme. Second barrier is the corona radiata cells. So for that, the sperm has the enzyme, which is a corona penetrating enzyme. Third barrier is the zona pellucida. For that, the sperm has the enzyme zona lysine. And this whole phenomena is known as the acrosomal reaction. Now, when the sperm come in contact with the plasma membrane of the secondary oocyte after crossing these three barriers, first thing happen is the fast block. Fast block occurs due to the depolarization of the plasma membrane of the secondary oocyte. Because of this depolarization, what happened? The other sperms, they cannot enter or penetrate into the secondary oocyte. So this block, it is blocking the polyspermy. So this block is fast, but it is temporary. Now what happens? After the first block, the cortical granules which are present, these are the cortical granules which are present in the cytoplasm of the secondary oocyte, they start coming close to the plasma membrane of the secondary oocyte. And these cortical granules, they start releasing the calcium carbonate in the perivitelline space, which is present between the zona pellucida and the plasma membrane of the secondary oocyte. So they start releasing the secretion of calcium carbonate in the perivitelline space. And these calcium carbonate start depositing on the zona pellucida. Because of this deposition, what happened? The zona pellucida becomes hard. Now no more sperm can enter into the secondary oocyte. This block is known as the slow block, and it is a permanent blockage to the polyspermy. Though it's slow, but it's a permanent blockage. And this is also known as the cortical reaction because cortical granules are involved. Now, when this sperm it enters into the cytoplasm of the secondary oocyte, it contains three things. Very important point, student. Remember, it contains three things. Number one, it contains APC, which is an anaphase promoting complex. Number two, it contains the haploid nucleus. And number three is the proximal centriole. So when the sperm enter into the cytoplasm of the secondary oocyte, it contains three things: APC, haploid nucleus, and proximal centriole. Remember, important point, that secondary oocyte, it's still arrested in the metaphase 2 of meiosis 2. That means the meiosis 2 is not completed yet, right? So meiosis 2 is completed when there is a presence of sperm. And why it is completed when only in the presence of sperm? Because the sperm carries this anaphase promoting complex. When this anaphase promoting complex reaches to the second cytoplasm of the secondary oocyte. Secondary oocyte completes the meiosis II. Now, when it completes the meiosis II, it forms the ovum and the second polar body. The polar body degenerates or die off again. This ovum nucleus of the sperm and forms the zygote. Right? Now you must be thinking, ma'am, then what is the role of this proximal centriole? Try to recall, student, that in meiosis or in cell division chapter, you have studied that all the cells except the ovum cell does not have the centriole. Remember this very, very important point. Ovum cells, they does not have the centriole. Now, once the zygote is formed, this zygote has to undergo the mitotic division to form the embryo. And for the mitotic division, it requires the spindle formation. And for spindle formation, it requires the centriole, right? Then how it going to get the centriole? So that centriole is contributed by the sperm. Remember, the sperm, it carries the two centriole, distal and proximal. Distal is involved in the tail formation. Proximal is the one which is involved in the cleavage or the mitotic division of the zygote. Now, once the zygote is formed, this zygote undergoes the mitotic division, and these mitotic division are known as cleavage. So the zygote will undergo the 2-cell, it will undergo the mitotic division, it will form the 2-cell, 4-cell, 8-cell, 16 daughter cells, which are known as blastomere. Now the embryo with 8 to 16 blastomere is known as morula. Morula is a solid ball-like structure. And it is formed in the fallopian tube. Remember the site, very, very important, that it is a solid ball-like structure and it is formed in the fallopian tube. Now this morula, it further undergo the division and transform into the blastocyst. Blastocyst is the one which enter into the uterus. That is why I'm mentioning that morula is formed in the fallopian tube, so that you people have a clear idea. Okay, morula is formed in the fallopian tube. Then this morula undergo the mitotic division and form the blastocyst. Blastocyst is the one which enters into the uterus. Now these blastocysts, they undergo further divisions and they form and the cells of the blastocyst, some cells of the blastocyst, they are arranged at the periphery, which are known as trophoblast. And some cell of the blastocyst, they are confined to one region or the one portion, and those cells are known as the inner cell mass. So remember, the inner cell mass is the one which is involved in the organ formation of the embryo. Trophoblast is only involved in producing the hormone and in the placenta formation. Remember this very, very important point, student, that the trophoblast is not involved in the organ formation of the embryo. It is the inner cell mass which contribute, because inner cell mass has the totipotent cells. It has the stem cell, not the trophoblast cells. And in the blastocyst, because one cells are arranged at the periphery, some cells are arranged at one portion. So there's a cavity is formed in the center. And that cavity is known as the blastocyst. So that cavity is known as the blastocoele. And remember that which stage of the embryo is implanted or attached to the endometrium of the uterus. It is the blastocyst stage. Very, very important. Remember the stage that which attached to the endometrium wall of the uterus. It is the blastocyst stage. And one more important point which I want to mention here, that when the blastocyst it enters into the uterus, on the day 5 of the fertilization, like on the day 5, hatching of the zona pellucida occurs. Okay. Hatching of the zona pellucida means that the zona pellucida breaks and the blastocyst will come out. Then you people must be thinking that why this hatching of the zona pellucida occurs only on the day 5? Why not on day 3, day 2 or 4? It because student, zona pellucida, it prevents the ectopic pregnancy. Ectopic pregnancy means the premature attachment of the embryo to any site of the female reproductive system other than the normal uterine site. For example, if the zona pellucida hatched on the day 3, let's say, and when the embryo is in the fallopian tube, so the embryo will attach to the fallopian tube, and you know that the fallopian tube, it has a very narrow lumen. So if this embryo will undergo the mitotic division, the size of the embryo will increase, and if this increased size embryo will start putting the pressure on the fallopian tubes, on the walls of the fallopian tube, and because of increased pressure, the fallopian tube will get ruptured. These type of pregnancy are known as the ectopic pregnancy. So to prevent these ectopic pregnancy, zona pellucida, it hatched on day 5, so that this blastocyst will attach at the normal site in the uterus. When the blastocyst attached to the endometrium of the uterus, this is known as implantation, and it leads to pregnancy. Now, one more important point which I want to mention here. Let's say this is the endometrium, and this is the blastocyst. Now, when the blastocyst or the embryo get attached to the endometrium, now we call endometrium as decidua. So this portion of endometrium will be known as decidua basalis. Now, part of the endometrium which covers this blastocyst, this is known as decidua capillaris. And part of the endometrium which are not involved in the implantation, those part of the endometrium is known as decidua parietalis. Remember this very, very important point that after the implantation, now you call the inner layer of the uterus as decidua. Now you no more call the inner layer of the uterus as endometrium. After the implantation, it is called as decidua. Remember this. I hope it is clear to everyone. It's already 11. We are winding up the class. Next part we'll do in the next class, okay? I hope this lecture was informative and interesting for you all.